In this module, we will provide an overview of the MEG signal generation. During the course of this presentation, we shall provide insight into how MEG tracks brain activity, what neuromagnetic fields are, why MEG has better spatial resolution than EEG, what pyramidal cells are and how they generate magnetic fields, why MEG is more sensitive to magnetic fields associated with tangential sources, and what factors affect the strength of the MEG signal. First, a brief review of electromagnetism. Try to remember your high school physics class. The Danish physicist and chemist Hans Christian Ørsted observed the deflection of a compass needle during an experiment and subsequently identified the direct relationship between electricity and magnetism. His subsequent experiments in 1820 showed that an electrical current flowing in a wire generates a circular magnetic field with the wire at its center. Often referred to as the right-hand rule, with the current flowing to exit the tip of the right-hand thumb, the field lines curl in the direction of the fingers of the right hand. This is the fundamental principle upon which multi-channel MEG field map interpretations are based. MEG and EEG track electric brain activity by measuring the electromagnetic fields generated by neurons. Due to conservation laws, any current flow in the head must form a complete circuit. In Ørsted's experiment, his wire was connected at each end to the positive and negative terminals of a battery. In our neuronal circuit, the primary current, shown here in red, flows in the postsynaptic neurons. The return current is distributed everywhere else in our volume conductor, shown here in yellow. Thus, an electric current generated by the cellular current in an active population of neurons generates both a magnetic field and an electric potential field, which extends extracranially and can be detected by appropriate sensors. In the case of EEG, potential differences are measured between two electrodes applied to the scalp. However, the skull is a poor conductor and the electric signals are distorted. In the case of MEG, changes in the magnetic flux are detected by magnetic field sensors outside of the head. Those fields pass without being distorted by the skull, making source localization in MEG more accurate than EEG. Let's look at these neural currents and fields in a little more detail. There are a few terms which you will hear associated with neural currents and fields which are important to understand. Impressed currents, shown as orange arrows, are ionic currents through open ion channels in the cell membrane. Primary currents, shown as red arrows, are the net intracellular current in the source area driven by the impressed currents. Return currents, also called volume currents, shown as yellow arrows, are the extracellular currents that close the electric circuits. They are considered to be driven by the primary currents, and they are present throughout the entire conducting medium, that is, the head. Although we illustrate these concepts on a single neuron level, they are applicable at macroscopic scales. Many thousands of postsynaptic potentials are acting simultaneously, and MEG and EEG sensors observe the activity at some distance from the neurons. As we know, all currents generate a magnetic field, and in our volume conductor model, they also generate an electric potential field. The skull is a poor and inhomogeneous conductor of electricity, so it distorts and blurs the electric potentials on the scalp generated by the neural activity. Fortunately, the skull has much less impact on the corresponding magnetic fields. The primary currents are directly related to neural activation, and we are seeking to estimate the locations of these currents based upon the measured MEG and EEG signals. When we describe the actual currents present in axons and dendrites, we broadly classify them as two types. Free synaptic action potentials propagate very fast and generate rapidly decaying magnetic fields. In addition, free synaptic activation has little or no temporal summation. Thus, the presynaptic activation contributes very little to the MEG signal detected outside of the head. After the activation propagates over synaptic clefts, postsynaptic potentials and currents, shown as the red arrow, are slower, much longer lived, and act over longer distance. These postsynaptic currents are considered to be the main source of the measured MEG signals. The primary current in a single cell and its associated magnetic field are far too weak to be detected by MEG. Fortunately, neurons do not act individually, and many thousands of co-located neurons will fire simultaneously to perform an action. The apical dendrites of the pyramidal neurons are largely oriented in parallel. If they were randomly oriented, the magnetic fields generated by the neurons would cancel each other. Due to this specific architecture of the pyramidal neurons in the grey matter, the summation of their activation results in signals strong enough to measure with MEG. Next, we will estimate how large a patch of cortex is activated to generate detectable MEG signals. From a distance, a postsynaptic current looks like a current dipole. Its strength Q is the product of the primary current I and the distance d over which it flows. The dipole moment unit is ampere meter, AM. In a single cortical neuron, the distance d is approximately 0.2 millimeters, and the primary current is approximately 100 picoamps, resulting in the dipole moment Q of about 20 femtoampere meters. Generally, measurable MEG signals at some distance from the cortex require the dipole moment strength of at least 10 nanoampere meters. This means that about 1 million synapses need to be simultaneously active to generate a measurable MEG signal. The density of pyramidal neurons is about 100,000 neurons per millimeter square, each of them having thousands of synaptic connections. Thus, as few as one synapse in 1,000 over an area of 1 millimeter squared would suffice to produce a detectable signal. In practice, there is partial cancellation of the generated electromagnetic field due to currents flowing in opposite directions in neighboring cortical regions. Therefore, it is estimated that approximately a 40 millimeter square patch of cortex activation corresponds to 10 nanoampere meters net dipole moment. The cerebral cortex is a 4 millimeter thick sheet of grey matter folded in a complicated way. As was shown earlier, the apical dendrites of cortical neurons are oriented parallel to each other and perpendicular to the cortical surface. They are shown here by the small yellow arrows. Because of the folding of the cortex, the net current in a sulcus, shown as the large yellow arrow, is oriented parallel or tangentially to the scalp surface. Corresponding net activation in a gyrus would be oriented perpendicularly or radially to the scalp. The green circles indicate the magnetic field generated by the neural activity in the sulcus. We will see in later slides how the orientation of the current matters. MEG sees mainly tangentially oriented currents in sulci, while EEG sees both tangential and radial currents. In the next few slides, we will have a look at the main factors that affect the strength of the MEG signal. First, the amount and type of synaptic input affect the signal strength. Excitatory synapses are arranged near the superficial cortical layers, and inhibitory synapses are close to or in the soma of the neuron, and they both generate neuromagnetic fields with similar orientation. Generally speaking, the excitatory synapses generate larger fields than inhibitory ones because the current flowing along a longer linear path will generate a larger field. Remember, the dipole moment Q is equal to the current times the distance, so the longer travel of the excitatory path generates stronger signal than the shorter travel of the inhibitory path. The degree of synchronization within the local neural population will also affect the strength of the generated signal. Next, the orientation of the neural current affects the MEG and EEG signals. Besides primary currents, return currents also generate a magnetic field. In the case of a spherical volume conductor, the magnetic field due to return currents will exactly cancel out the field due to any radially oriented primary currents. Therefore, all radially oriented currents, shown here as yellow arrows, will not generate a magnetic field in a spherical head model. In a more realistic head geometry, they will generate small MEG signals, but much weaker than those from tangential primary currents, shown here in orange arrows. Usually, the neural activation causes obliquely oriented primary currents, shown here with red arrows. In these cases, the tangential current components generate most of the neuromagnetic fields, with small contributions from the radial current components. This is demonstrated in this illustration of the source strength needed for detection across the cortical surface. Besides the orientation, the source depth has a major impact on the strength of the MEG and EEG signals. When the primary current is located deeper and deeper from the surface, the signal is progressively cancelled by the return currents. In fact, in a spherical conductor, there is perfect cancellation of fields at the center of the conductor. Furthermore, opposing primary currents in close proximity will partly cancel each other, thus reducing the detected signal amplitude. To summarize, MEG signals recorded at the scalp are generated by synchronous activity of tens of thousands of neurons. Mainly, postsynaptic currents in apical dendrites contribute to MEG signals. MEG signals are highly sensitive to currents tangential to the skull originating in the cortical sulci. MEG fields reflect the primary neuronal currents directly, with minimum distortion from different layers, brain tissue, skull, scalp, with different electric conductivities. Estimation of MEG sources implies the construction of computational models of the biophysical sources and of the volume conductor. The manuals that come with your MEG system contain important information about using your MEG, including safety warnings. You must familiarize yourself with this material before operating your MEG.